just double check my screen share. Okay. And that looks good on your end? Perfect.
Okay, looks like we are live streaming on YouTube, and I don't know if we asked you in the emails, are both of you okay with this talk staying up online as part of our archive, or do you want it taken down after people get a chance to view it?
It should be fine being up. So we just submitted a paper on all this research, so.
Congratulations, that's great. Yeah, a lot of people who either can't make it live, catch up later, and then also people return to the resources over time just to, yeah. I mean, I've been using these talks as a huge resource in preparing the class that I've been teaching. So it's been super helpful. All right.
Let's see how many of the European crowd comes today, because I'm guessing there's a meeting going on.
Yeah, I've been seeing on Twitter lots of chatter about everyone's so excited to be doing their first in-person meeting in the UK. We're all sitting over here super jealous. I think there's actually a meeting in the US too. There's a lot going on.
Everything is starting back. Yeah.
Yeah, things are starting back up. That's exciting, but hopefully people still want to come together in this format. Okay, so in the interest of time, let's go ahead and get started. So hello, everybody. Welcome to another exciting installment of Cell Migration Seminar Series. So today we're doing something a little bit different where we have a joint talk. We'd actually been in Is my microphone still working? Sorry, I got one of those errors, Zoom saying it stopped working, of course. We've been in conversation with Professor Adam Engler from UCSD, and he said to us, actually, he wanted his trainees to get a chance to present their work instead. So we really appreciate that. That was great. And so today we have a joint talk between Katherine Birmingham and Benjamin Yeoman from UCSD. First, we'll have a talk from Ben, who works with Adam Engler at UCSD, but he's also a joint PhD student with Parag Katira at SDSU. And then we'll take some questions, and then we'll have a second part of the talk from Katherine Birmingham. So she is a postdoc in the Engler lab. She completed her PhD in 2020 in mechanical engineering and bioengineering at the Georgia Institute of Technology in the lab of Dr. Susan Thomas. So mad props to you, Katie, for finishing your PhD at such a tough time. And with that, I will hand it over to both of you. And I think, Ben, you're going first. So go ahead and share your screen, and the floor is yours. Thanks so much.
All right, thanks, Jennifer, for the introduction. This is research both Kate and I have been working on for the last couple of years now. And so what we're trying to understand is how cell adhesion plays a role in cancer metastasis, and whether or not we can use that as a prognostic marker for metastatic potential. So cancer, it remains one of the leading causes of death in the United States. But most of these cancers are due from carcinomas. So these are cancers that arise from epithelial cells. They typically form solid tumors, but between tumors of different origin, you don't really have much genetic similarity. So our idea is using something like cell adhesion or some physical marker to actually diagnose how aggressive a certain type of cancer will be. And so a lot of my work focused on the top three most diagnosed cancers, so lung, breast, and prostate. Cancer metastasis is what's really responsible for the high mortality of these carcinomas. So it's the process of primary tumor formation, vascularization, detachment and invasion, and then subsequent tumor formation. So as soon as secondary tumor forms, the 5-year survival rate for patients substantially decreases. So our research, we're really looking into the role of how these mechanical properties allow cells to detach and invade in the first place. But the big issue is even within a tumor, there's not a lot of genetic similarity. So you can have these heterogeneous populations where some cells are more or less adherent than others. And so these biophysical markers, they're really emerging as metrics for metastatic potential. So things like cell stiffness, cell traction forces, and especially cell ECM adhesion. So our idea is that even though these cancers might have differences in their genetic makeup that causes them to become cancerous in the first place, our thought is that these mutations might converge into similar phenotypes, much like how bats and birds are different species, but they use the same mechanics to fly. Our thought is that maybe these cancer cells actually use the same mechanics to escape the tumor in the first place, which could be a more universal marker for cancer risk and potentially a new target for new therapies. So we developed a parallel plate flow chamber where we can isolate cells based on their adhesion strength. So these cells, they're seated onto an ECM-coated glass slide where we can then shear them at a low flow rate and detach any weakly adherent cells, and then increase the flow rate to wash away all the moderately adherent cells and then trypsinize to collect the strongly adherent population. We can then see these cells onto hydrogels, time-lapse image them, and then measure how quickly they migrate. And so what we find is that when we use a non-metastatic MCF10A breast epithelial cell line, we don't see any differences between the weakly and strongly adherent fractions. But a cell line with just the HRAS gene transformed, which makes them more tumorigenic, we find that weakly adherent cells become more migratory than their strongly adherent counterparts, which is exactly what we see in breast, lung, and prostate cancer cells. So all the weakly adherent fractions migrate faster than the strongly adherent cells. So it suggests that this might be something that's more universal across different types of cancers. And so we wanted to see if this was also true in 3D. So we took the breast cancer cells, the MDA MB231 cells, and formed spheroids from these cells to do a 3D dissemination assay. So once we formed the spheroids, we embedded them in a 3D collagen gel, and then just monitored how far either weakly or strongly adherent cells spread after 24 hours. So the maximum displacement, it was the same across all groups, so weakly, strongly, and unselected. But the final area divided by the original area was much higher for the weakly adherent cells compared to the strongly adherent cells or even the unsorted group. So it seems like even in 3D environments, the weakly adherent cells are able to migrate more quickly than the strongly adherent cells. So we then looked to find what might be driving these differences in adhesion strength. So we ran a Western blot and stained for some typical focal adhesion proteins, and didn't really see any differences between the strongly and weakly adherent cells. So we thought maybe it has to do with how quickly these cells are actually disassembling these focal adhesions and reassembling them to migrate. So we did this assay where we imaged the focal adhesion of weakly and strongly adherent cells in cation-free media as well as cation media. So the cations, they activate the integrins, make them bind more tightly to the ECM. So when you remove them, these bonds become less adherent and the cells will quickly disassemble their focal adhesions. And what we find is that when we remove these cations, the weakly adherent cells disassemble their focal adhesions much faster than the strongly adherent cells. So once these cations are removed, you have a much fewer focal adhesions per cell area, as well as focal adhesions, number of focal adhesions divided by the cell, normalized to the cell area. So the focal adhesions, they're smaller, as well as fewer of them per, or normalized to the cell area. We also find that weakly adherent cells are more contractile than the strongly adherent cells. So we perform traction force microscopy, where we culture the cells on 2D polyacrylamide gels embedded with fluorescent microbeads. So we could measure the displacement of these beads before and after the cells contract. So we'll trypsinize the cells, wash them off the gels, let the beads relax, and then measure how strongly they're pulling against the polyacrylamide gel. And what we find is that weakly adherent cells in the tumor microenvironment, or stiffness that matches the tumor microenvironment, are more contractile than their strongly adherent counterparts. So what we think is that because these cells are generating more forces on these focal adhesions, maybe that greater force allows them, or there's more strain on that bond to begin with, so they're more likely to break than with a less contractile, strongly adherent cell. We then did RNA sequencing, and we found that there's 500 differentially expressed genes between the weakly and strongly adherent cells. And so you can see there's quite a few that are differentially expressed between the weakly adherent and the strongly adherent, either upregulated or downregulated. But what we find is that many of these upregulated genes are involved in microtubule and cytoskeletal organization, as well as binding. So some of the gene ontology terms really have to do with microtubule, cytoskeletal elements, some motor activity, which I think is leading to some of the differences in contractility. And then a lot of transport proteins, transport motor proteins that might be pulling away some of these focal adhesion proteins at the leading and trailing edges of the cell. And so a lot of these genes here are also involved in some processes that have to do with cancer metastasis and things like that. So we ran a PCR validation just to make sure that what we saw from the RNA sequencing is actually real. And indeed, a lot of the genes are upregulated in the weakly adherent cells relative to the strongly adherent. And so we wanted to validate this by targeting some of these microtubules. So using nicotazole to destabilize them, and then paclitaxel to stabilize microtubules. In both cases, the weakly adherent cell speed decreased when we treated these cells, whereas with the strongly adherent, we didn't see any difference in their migration speed. So this really suggests that these genes that are regulating the microtubules might have something to do with the differences that we see in cell speed. So we then, using the Cancer Genome Atlas, compared the differentially expressed gene profile of the weakly adherent cells to what we see in the Cancer Genome Atlas, using patients that were diagnosed with triple-negative breast cancer. And what we find is that patients that have a similar profile to the weakly adherent cells tend to have a shorter progression-free interval, as well as a shorter disease-free interval compared to the strongly adherent cells. So these genes that are driving this also show up in real patients and have a significant outcome with those patients. So getting into more of how these cells are behaving in the tumor microenvironment, we wanted to see how they migrate in response to stiffness gradients. So as you have primary tumor formation, the ECM surrounding that tumor becomes stiffer relative to the surrounding stroma. And typically healthy cells will durotax, so they'll migrate from softer environments into stiffer environments. And you see this almost universally across 2D migration assays and 3D migration assays. Cells always tend to migrate towards the stiffer environments. But in cancer, these cells need to migrate opposite of that. So from the stiffer tumor through the softer stroma to eventually get to the bloodstream. So this suggests that whatever mechanisms regulating durotaxis and metastasis must be disrupted in these cells in some way, or our understanding of how durotaxis works isn't entirely understood. So we made these photopattern gels that mimic the tumor microenvironments. We have these alternating soft and stiff regions, and we tune it so the soft regions match the stroma and the stiffer regions match the tumor for the specific type of cancer that we're using. We can then see the cells on there and then watch them migrate over 24 hours and measure their distance to their closest boundary. So whether that's stiff or soft, we just get the relative distance to whatever boundary they're closest to. And we can use that to see when they cross over from stiff to soft or soft to stiff. And what we find is that strongly adherent cells in the breast and prostate cell lines that we used were more durotactic than the weakly adherent cells. So if they started in the soft regions, they were more likely to end up in the stiffer regions after 24 hours. And you can see this in the population distribution. So when we initially seeded the cells, it was pretty evenly distributed across the soft and stiff. But after 24 hours, there's this distinct shift of the strongly adherent cells into that stiffer region, whereas the weakly adherent remained relatively evenly distributed. So it suggests that the strongly adherent cells are much more durotactic than the weakly adherent cells. So like I mentioned earlier, the weakly adherent breast cancer cells are more contractile, but we also see that with the prostate cancer cells in the tumor microenvironment. But these weakly adherent cells are also more contractile in the tumor compared to the stroma, and likewise with the strongly adherent cells. So as you increase the stiffness of the environment, the cells become more contractile, and the weakly adherent cells tend to be more contractile overall than the strongly adherent cells. So we think integrin bond dynamics might be playing a role in some of this divergent durotactic behavior. And traditionally, these bonds have been thought of as slip bonds. So as you increase the force on the bond, the lifetime decays exponentially until the bond breaks. But a lot of new research shows that they actually behave more like catch bonds. So they decay to some minimum before increasing again to some optimum bond lifetime at some optimum force, and then decay again until they detach. So much like those finger trap toys where if you pull them softly, you can get your fingers out, but if you pull too tightly, they tighten down and that lifetime increases. So the way this could work is a cell that is moderately contractile. It's going to generate lower forces in the soft region compared to the stiff, and also have a lower bond lifetime compared to the stiffer environments. So the forces that are generated by all these stress fibers on the cell are going to add up in a way where that net force will push that cell towards the stiffer environment. But a more contractile cell, you can actually shift the bond lifetimes. So the bond lifetimes will be longer in the softer environments compared to the stiffer environments. And so even though the forces are smaller, you could have more bonds that form in that softer region compared to the stiffer region. And with more forces acting on the cell towards the stiffer direction, those forces may balance out, making the cell ageotactic. So what we see with the strongly adherent cells, they're less contractile than they are in the soft regions compared to the stiff regions. But when you increase their contractility overall, you can effectively reverse these bond lifetimes in the soft and stiff environments. So we used a spinning disk adhesion assay to measure whether or not adhesion strength actually changes on these gels. So we coated a glass cover slip with a polyacrylamide gel to either the stiffness of the tumor or the stiffness of the stroma. Spun this disc in PBS and then measured the detachment from the radius to the center. So higher shears near the edge, whereas they're close to zero at the center. We can generate an adhesion profile, figure out what the median shear of these cells are. And then what we find is that the median shear to cause half the cells to detach was much lower for weakly adherent cells in the stiff environment. So with this catch bond lifetime, we'd expect that strongly adherent cells could be spanning this hump here, whereas the weakly adherent cells in the stiffer environments are much more contractile, so those bond lifetimes end up being reduced. So we then built a computational model to kind of simulate how these cells might be behaving. So each cell, it has some number of stress fibers. Each stress fiber is given a certain number of myosin motors. Those motors will then transmit that force through a stress sensor protein and an adapter protein, where the stress sensor can recruit more stress fibers. These then bind to an integrin, which attaches to the ECM. So by generating a force from that stress fiber that's dependent on the ECM stiffness, we can get a force that will determine the off rate of the integrin ECM bond. So they can then simulate how these cells migrate over these gradient gels over 24 hours with different contractilities for the cells. And what we find just by changing the maximum force these cells can generate, the less contractile cells behave much like our strongly adherent cells, where they're more durotactic, and you see that same shift in population after 24 hours. So we wanted to see if we can actually titrate this durotactic behavior. So we decreased the maximum cell contractility by 5 piconewtons, and find that as we decrease it, the durotactic behavior increases. So making the cells less contractile made them more durotactic. We then tested this experimentally. So using blebbistatin to inhibit non-muscle myosin 2. We found that as we knock down those myosins, making the cells less contractile, we can restore the durotactic behavior in these weakly adherent cells. And then with the strongly adherent, we increase their contractility using LPA, which is just an agonist to increase cell contractility overall. We find that it knocks down their durotactic behavior after 24 hours. So to make accurate measurements of overall cell adhesion strength and develop a easy to use assay for measuring this adhesion strength, we designed this parallel plate flow chamber that has this divergent channel within it. So along this path, the wall shear stress that the cells experience is going to increase exponentially, and we can measure this nice adhesion profile of the cells that we're interrogating. And that gives us the tau 50 value that we use as a metric for where 50% of the cells detach at some given shear. And so we looked at a panel of different breast cancer cell lines. So some more metastatic than others, and then a couple normal breast cell lines. And what we find is that the lower the tau 50 of these cells, so the lower their median detachment is, the higher or the faster they migrated, and the further away they migrated from their starting position. So this suggests that more metastatic cells, they are more migratory and have a lower adhesion strength compared to less metastatic cells. So what's in green is our cell lines that have been characterized to form secondary tumors in mice, whereas what's in blue typically don't form secondary tumors. So there's a nice cutoff between the tau 50s of more metastatic and less metastatic cell lines. But the problem with using this as a diagnostic tool is when you take a patient sample, you're not just going to get cancer cells. You're going to get a mix of both cancer cells and healthy cells in the sample. So we wanted to see if we could distinguish the two populations from within the single adhesion profile that we get. So we set up this experiment where we mixed non-metastatic MCF10A cells with metastatic 231 cells. So we cold cultured them and then sheared them in this device and measured their overall adhesion profile. So you have an initial drop off where the less adherent cells detach, and then a second one where the more metastatic cells detach. So we use this equation here. So it's a sum of two vivo functions. So they just generate a single survival curve. So we sum those up together and fit it to extract the cancer fraction, so the number of cancer cells that are in that sample, and the tau 50 of those cancer cells, knowing the adhesion profile of the more adherent cell line. So if we know the tau 50 or the parameters that generate the adhesion profile for the more strongly adherent cells, we can extract the adhesion profile of the less adherent cancer cells. So measuring this experimentally, we have the adhesion profiles of the cancer cell line and the healthy cell line. With the cancer cell line, cells detach sooner than the more strongly adherent healthy cell line. And so with the cold culture, you can see where the cancer cells start to detach and then where the tumor cells or the healthy cells start to detach. So we can fit this and then predict what the seeded fraction was, seeded percentage. And it's pretty close to what we had initially seeded. And then the tau 50 of these predictions was within plus or minus 40 dynes per centimeter squared to what the actual tau 50 for the 231 cell line was. And so with that, I'll conclude with the weakly adherent cells. They tend to be more migratory, more contractile, and don't durotax the same way that their strongly adherent counterparts do, which suggests that these weakly adherent cells are more metastatic to begin with. RNA sequencing of both weakly and strongly adherent cells suggests that these differences in adhesion are driven by cytoskeletal components involved in migration and locomotion. And patients with similar DEG profiles to strongly adherent cells typically had better outcomes. And so our device, it can accurately distinguish the percentage and median adhesion strength of cancer cells within these mixed populations. So we think adhesion strength using our device could be a good way to prognosticate how aggressive a patient's cancer will be. And so before I pass things off to Katie, I'll take any questions.
Okay, great. Thanks, Ben. If anyone has questions, feel free to drop them in the chat or use the raise hand function. I just had one about, well, I have actually a bunch, but I was wondering about the spheroids that you made when you have between your strongly adherent and your weakly adherent cells. I was just curious if they behaved any differently. Did you see your weakly adherent spheroids kind of falling apart, or did that not really happen because they still had kind of, enough expression of junctional molecules to keep them together, even though they were, not as adherent in a flat culture?
With 231 cells in general, they typically don't form a lot of cell-cell junctions. So I remember both of them being pretty fragile. They're difficult to work with, but they held together well enough where we could get most of them embedded in the collagen.
Yeah, okay, cool. That makes sense. And then I was also just wondering, so after you do your initial selection where you have these, your strongly adherent, your weakly adherent populations, presumably then you sort of kept culturing those in flasks and, you know, sort of kept those going as splitting. And I was just wondering if there were, you know, were you also selecting for morphological differences? Were there cell shape differences? Could you tell just, you know, if somebody showed you a flask, which one it was, or did they still look the same afterwards?
They weren't that much different. The weakly adherent cells might have spread less. So their cell area was a little bit smaller, but unless you measured it quantitatively, it was hard to tell just by looking at them.
Yeah, okay, that's interesting. Yeah, Ankita, please go ahead.
Thank you so much for the wonderful talk. I was thinking more in terms of the complexity, you know, tumors usually have, like, it looks like somehow the contractility is being modulated. So how do you picture maybe things like confinement or crowding in the tumor have an effect on that rather than actual durotaxis, you know, moving from a tumor to a, like, how does other ECM components must be? And I'm maybe I will add to that. I'm asking that because, you know, reading a bunch of papers where they talk about an upregulation of maybe integrins in highly like metastatic cancer cells.
Mhm.
You would think that they will probably make more adhesions, but it looks like they do less.
And yeah, fully recreating what's going on in the tumor is pretty difficult. I'm sure crowding has some effect as well as different chemotactic agents. So
Yeah,
the blood vessels or the endothelial cells in the blood vessels are secreting something, then maybe the cancer cells are responding to that.
so I'm just wondering how much of like local changes in stiffness do they really see in a tumor versus, let's say, the neighboring.
And what they've found in tumors is that the gradient between the stiffness of the tumor to the stroma, I think it's between like 10 pascals per micron to 100 pascals per micron.
Mm-hmm.
So we tried to recreate what was pretty close to that in our system.
Great. Okay. Thank you so much.
Great, thanks. So Frank had a question, but it looks like it was answered by Adam about the longevity of these phenotypes. So I'll ask the next question from Nile Tinjaya, saying interesting work, and they're wondering if you had made any direct tension measurements on the weakly adherent versus the strongly adherent cells, because it was interesting that the weakly adherent cells generated higher traction forces. Agreed. And they would imagine that they would migrate faster, but just not deform the substrate as much.
So we didn't do any direct tension measurements. The cells were just measured on the polyacrylamide gels with the fluorescent beads. So we didn't measure like the floors at a single focal adhesion. We don't really have in our lab, we don't really have the capabilities to take that high of resolution. I think it'd be interesting to look at, especially if we could do it as the cells are crossing from soft to stiff, like spanning that boundary. And I think from the model though, it suggests that just the way these catch bonds are behaving differently based on the stress generated from these stress fibers is enough to explain some of these differences that we see.
Great. Next question is from Ernest Liu asking, when the strongly adherent cells durotax, do they switch from less contractile to more contractile? And there is a second part of the question. How do you know that these cells are still strongly adherent when stiffness of the environment is changed?
So the way we define strongly and weakly adherent, it's done on glass slides. So the cells are already on the maximum stiffness that they'd experience. Stiffness or adhesion does change based on the substrate stiffness. So getting into actually using this as a prognostic tool, I think as long as the cells on glass are enough to make good predictions that the stiffness in these measurements shouldn't matter too much. But then as far as the changes in contractility when they switch between soft and stiff, this has been pretty well characterized in a lot of experiments where they measure attraction forces across different stiffnesses. And cells just tend to be more contractile as you increase the substrate stiffness. So there's not like one contractility mode they have across all stiffnesses. It changes based on what they're experiencing.
Thanks, Ben. So I think we'll just take one more question and then we'll switch over to Katie. So from Frank Charbonnier, asking, for cell types that form stronger cell-cell adhesions, do you see any relationship between cell substrate adhesion strength and cell-cell adhesion strength?
So when we typically measure the adhesion strength, we see the cells at a low enough density where they're not interacting with each other. When we do have too many cells that are kind of aggregating, then the measurements aren't as accurate, just because I think there are some cell-cell interactions causing some of that error.
Okay, great. Thank you so much. So I think if you can stop sharing your slides, and then Katie can go ahead and bring her talk up.
Okay, great. Give me 1 second to bring the slides up.
Yeah, no problem. And then at the end, obviously, people can continue to ask questions to both of you. So if more questions come up for Ben, I'm sure we'll make time for that as well. So with that, Katie, please take it away. Floor is yours. Thanks so much.
Okay, great. Can you guys all see the slides? Okay. Great. So Ben did a really great job explaining all of these in vitro differences that we saw between our strongly adherent and weakly adherent cell subpopulations. So I'm going to talk about an extension of the work where we then took these findings and applied them to different in vivo experiments to further look at differences in invasiveness and metastatic potential of our different adherent cell subpopulations. So first, we were interested in looking at if these in vitro migration differences correlated with differential invasiveness in vivo. And so in order to perform this experiment, we took these MDA MB231 cells that Ben talked about previously and transfected them with a GFP and luciferase. And so this allowed us to see these cells under fluorescent light and then also image the mice using an IVIS machine to look at the tumors. And so we injected NOD-SCID mice with 1 million MDA MB231 cells. We then let the tumors grow in the mice for 6 weeks, after which we sacrificed the mice, resected the fat pad, and then separated our tumor from our stroma. We then minced the tissue to isolate the GFP cells using FACS flow cytometry, and then quantified the adhesion strength of cells taken from the tumor and also cells taken from the stroma. And what we found was that while there were more tumor cells or GFP positive cells within the tumor as compared to the stroma, the cells that were in the stroma had a lower average adhesion than the cells that were in the tumor microenvironment, indicating that these invasive tumor cells have this weakly adherent phenotype compared to cells within the tumor. So based on the results of that experiment, we next sought to determine if these in vitro migration differences correlated with differential metastasis in vivo. And so again, we took these transfected MDA MB231 cells. Now differently, instead of injecting an unsorted population as we did in the previous experiment, I separated these cells into a strongly adherent subset and a weakly adherent subset using the parallel plate flow chamber that Ben explained previously. I then injected NOD-SCID mice with 1 million weakly adherent, strongly adherent, or unselected cells into the mammary fat pad of the mouse. and then monitored the tumor growth for 6 weeks. After 6 weeks, I excised the lung and the tumor. And then first I performed mRNA sequencing on the tumors that were taken from the mice. And then I quantified the number of lung metastases that were in the tumor or the mice. And interestingly, I found that there was no difference in the size of the primary tumors between groups. So our mice injected with weakly adherent cells, strongly adherent cells, or this unselected population all had similar tumor growth when normalized to the weight of the mouse. However, I did find that there were transcriptome differences based on adhesion phenotype. So these primary tumors had this mRNA adhesion phenotype that profiled that correlated with the adhesion phenotype of the cells with which they were injected with. And when looking at the Go terms between these differentially expressed genes, I found that there were common Go terms between these in vivo mRNA sequencing data and then the in vitro mRNA sequencing data that Ben talked about previously. And specifically, there was overlap between biological processes and the cell component that were related to migration and locomotion. And then when looking at the number of metastases found in the lungs, I saw that the weakly adherent cells had significantly more lung metastases than mice injected with either strongly adherent cells or this unsorted control population. However, the size of the lung metastases did not differ, indicating that there was no difference in the method of the cell leaving the primary tumor microenvironment and traveling to the lungs, but rather it was a difference in the number of cells that were able to escape this primary tumor microenvironment. So next we performed an extension of this experiment where after injecting mice with either strongly adherent, weakly adherent, or unselected cells, at 6 weeks after sacrificing the mice, the tumor, stroma, and contralateral fat pad were resected. The tissue was dissociated and the detachment of these cells was assessed using the divergent parallel plate flow chamber. So again, this divergent parallel plate flow chamber allowed us to quantify the adhesion over a range of shear stresses. And using this data, we perfused the cells through the parallel plate flow chamber and then determined the average threshold of the cell population, so cells within the tumor or the stroma. And notably, the adhesion profile of the contralateral fat pad was used as the deconvolution standard to obtain the cancer fraction and average shear thresholds of the stroma and tumor samples. We found that in both the tumor and stroma samples, the number of lung metastases was inversely proportional to the measured average shear thresh of the cells. However, we found that only the stroma showed a positive correlation between the estimated cancer fraction and the number of lung metastases, thus indicating that the stromal tissue is a better predictor of disease severity than the tumor biopsies. So after noting these correlations, we next fit the number of metastatic lesions, average shear threshold, and cancer fraction with a logistic regression model based on the classification of high risk and low risk. So here, high risk is defined as a lung that had 2 or more lung metastases, and then low risk being a mouse that had 1 or fewer metastases. This model generated a probability score between 0 and 1 based on the likelihood of having a high risk tumor. We next use the probability estimates in a RAC analysis to evaluate how well the cancer fraction, and its corresponding average shear threshold classified metastatic risk. We found that this prediction model has a high accuracy as defined as this area under the curve of about 0.94, as well as a high specificity and sensitivity with percents of 185 respectively. So in conclusion, these in vivo experiments showed that invasive tumor cells exhibit lower adhesion than cells within the tumor. In vivo, these weakly adherent cells have a higher metastatic potential than strongly adherent or unsorted cells, even after early resection of the primary tumor. Further, we found that adhesion phenotype in vivo is correlated with a unique transcriptome profile. And adhesion of the stroma cells are predictive of in vivo metastatic tumor formation with high specificity and sensitivity. And so together, these experiments showed that adhesion strength and cancer fraction are two label-free metrics that can serve as markers of metastatic potential. And together, this kind of brings me to next steps and future applications of this divergent parallel plate flow chamber. And so we are in the process of exploring if this divergent parallel plate flow chamber can be used to predict clinical outcomes of breast cancer patients. And so this would involve acquiring samples of a patient tumor. So using a core needle biopsy to get tumor and stroma tissue, fractionating the tissue, and then isolating these epithelial cells using, again, FACS flow cytometry. We could then perform a diagnostic assay where we would shear the cells on the divergent parallel plate flow chamber, assess the percent detachment, so generate this average shear threshold curve, and from that create this risk phase diagram to determine if a patient might fall under a high risk primary tumor or a low risk primary tumor, thus giving clinicians a good metric for determining how to best treat the patient. So part of this study would also involve following patients that enroll in this trial for a number of years to look at their recurrence-free interval in order to assess the accuracy of our predictive model. And so with that, Ben and I would like to thank our lab, particularly our PI, Dr. Adam Engler, as well as Afshin, Pranjali, Jerry, Gabe, and Izzy, who are all key members and contributors to a lot of this work shown today. Further, from the Katira lab, we would like to thank Parag as well as Esteban. And with that, I will take any questions.
Thank you so much, Kathy. Let's wait, people take some time to trickle down some questions. I was again wondering about, how things get modulated. Like for me, it is very difficult to grasp the adhesion of the tumors when they start versus when they have metastasized and, reach their point. They have undergone these huge changes in terms of, let's say, when they're passing through different kind of microenvironment. So I don't know, I'm not able to grasp how good as a predictor adhesion is going to be, in these cases where they're able to, so because cells being plastic, they're able to modify that so quickly. Like, do you have some comment on that?
Right, so a lot of what we look at are cells within that primary tumor. So we haven't analyzed the adhesion phenotype of the cells that have escaped to the lungs, but we do see, going back, that there is clustering based on these cells taken from the primary tumor into their adhesion phenotype, at which point they are injected into the mice. So we see that 6-week 6 weeks later, there are some inherent differences that still remain between these strongly adherent and weakly adherent cells that might indicate that through mRNA sequencing, we would be able to profile if a cell has this strongly adherent phenotype or this weakly adherent phenotype.
Great, thanks. We have a question from Francisco Vega saying, great talks and data. And have you observed other phenotypic differences between the two cell populations besides adhesions, for example, stemness properties or other markers?
Yeah, so through our extensive mRNA sequencing that we did on both the in vivo and in vitro experiments, we did see again, hundreds of differentially expressed genes. So a lot of them correlated with migration and locomotion, but we also saw genes correlated with numerous other processes and classifications. So I don't know offhand all of the different genes that we saw that were differentially expressed, but there are many different genes corresponding to many different processes. And Ben, I don't know if you want to add anything to that.
Not specifically on stemness. I'd have to go back and check. But we did also measure cell stiffness, so using AFM. And the weakly adherent cells were slightly more stiff, I think, but the differences weren't really significant. I think we just had a lot of error in some of our measurements.
Yeah, I'm gonna go ahead.
Sorry, adding to that, I was wondering if there is something to do with the survival too. It's often thought that the most adherent cells have better endocases, but there are more and more data coming out that's probably not always true. So would you also think that there is something, like, did you find some data related to that in your RNA-seq?
So I don't know offhand if we saw any data related to that with our RNA-seq, but regarding the survival of the different cell populations in vitro, we do see that the cells grow at the same rate. And then also we saw with the size of the primary tumors that there were no differences. So that would suggest that we don't see any proliferation differences between the weakly adherent and strongly adherent cells.
Thank you.
You're welcome.
So when you're envisioning this as a diagnostic tool in the future, is the main idea that you think that you'll be able to profile the primary tumor and then think about, as a predictor to maybe tailor treatments and think about, how aggressive the tumor may end up being? Or is that right or no?
So I think we would actually profile the stroma. So when we looked at the correlation between resulting metastases and then also cancer fraction, when comparing cells derived from the tumor versus the stroma, we found that there was a better correlation of risk with stromal tissue as compared to tumor tissue. So I think the idea would be to take tissue from the stroma. However, once we begin this study, we would look and further evaluate the tumor versus the stroma correlations. But right now, our idea is to take stromal tissue.
Right, thanks, fantastic.
Wonderful. I think we have two questions, one for Ben and Katherine. One for Katherine. So I'll take Katherine's question maybe first. From Anna Pasapera asking, did you analyze different metastatic behaviors, including different tropisms to bone and lung that could be associated with discrete variation in gene expression patterns?
We did not. We only analyzed lung metastases.
Thank you. Next question is for Ben.
Yeah, from Nile asking, they're curious whether the mathematical model also accounts for the fact that as the cells undergo adurotaxis, they would also form fewer ventral stress fibers and more dynamic adhesions at the cell front. And then they have a related question, but I'll like answer that first.
So with the model, we didn't intrinsically program any differences for the cell's behavior on soft or stiff. It all had to do with the forces dependent on the stiffness. But we did add in where stress fibers can grow more quickly at the leading edge than the trailing edge of the cell. So with ventral stress fibers, where they're not necessarily bound to focal adhesions, we didn't include those. So just to simplify things, we just had defined a stress fiber as a force generating element that's bound to an integrin. That way we can model the directions of all the forces acting on the cell to then move it in a given direction.
Okay, great. And I think this is sort of a follow-up to that then, does the model only consider, and I think you've kind of answered this, but does the model only consider adhesions at the ends of the ventral stress fibers, because nascent adhesions, while they're weaker, are more related to protrusive activity?
So yeah, every stress fiber that we had in the model, it was attached to an integrin. Only stress fibers that were bound to integrins generated forces. And then we did include other integrins that would be located like somewhere in the cell membrane that act more as friction on the cell rather than any sort of force generating element. So just integrins that happen to be in the membrane but not bound to a stress fiber that generate friction on the cell.
Okay, thank you. And thank you. The question askers are saying thank you in the chat as well. So yeah, so I think that is all of the questions that we have. So I want to thank both of you so much for doing this. I think this joint talk format is really cool, and hopefully we'll get to do it a little bit more in the future. So thanks for spearheading this for us. And also thanks to Adam for putting both of you forward to give these fantastic talks. With that, I will end our official recording part. So to anyone